Yeah, so I want to thank HFSP for supporting this work, but also giving me the opportunity to share the work. And I'm going to switch from Golgi bodies to bacteria. And my lab's goal is, can we electronically control the behavior of bacteria by the structures which we call protein nanowires. And this is because our goal is, if I can move this forward. For this one, right? Okay. And this is because bacteria have an ability to solve several environmental problems. And two problems which we are focused on is how can we reduce the atmospheric methane released by this bacteria, and archaea, and also how can we target pollution using the bacteria. And the challenge is, can we manipulate this bacteria at a large scale and efficiently so that we can carry out these functions such as methane removal or bioremediation. And this is the scheme of our experiment, where we have been able to use this bacteria which produce these electronically conductive filament, which we call nanowires. And we can use tools like electric field, magnetic field, or a light to control the behavior of bacteria, and that controls the exchange of electron between them. And this is what eventually controls the gene expression. And using these protein nanowires, we have been able to control the bacterial metabolism, their ability to form communities like biofilm, and also the communication between these bacteria. So what I want to convince you today that it's all about these electrons produced by the bacteria. And this is the way these wires look like. So this is an image of a bacteria called Geobacter. Geo is Earth. And this bacteria was discovered not far from here in the Potomac River in DC. And it's one of the most common bacteria in our soil. And this is the electron microscopy image where you can see these hair-like filaments which function as wire. And these filaments, which I'm showing you here in the gray color, they have a continuous arrangement of heme molecule, and that allows them to move electrons over long distances. And there are a couple of surprises we found with this wire. The first is the electrons move at an ultra-fast rate. That means within 100 femtosecond. And we are trying to understand what allows them to move this electron over such a long distance. And the second surprise is the bacteria can move electrons over 10,000 times their size. And we are also trying to understand how are they doing that. And the reason why these bacteria make this wire is to survive without oxygen. So if we think about in our case, when we produce electrons and protons, by breathing in oxygen, we can get rid of all those excess electrons. But these bacteria are growing deep in the ground, where there is nothing like oxygen, which is soluble, membrane ingestible, which they can breathe in. So instead of taking oxygen in, they can actually export the electrons out. And this allows them to transfer them to minerals in the soil or a partner bacteria using these filaments made up of cytochrome. And we are targeting this process to control bacteria by controlling this electron transport. So let me show you how these wires actually look like. So on the top, I'm showing you a cytochrome monomer in different color. And they basically form a polymer. And each monomer is identical. It just rotated by 30 degrees. And at the bottom, I'm showing you the hemes within this wire. So you can think of them just like your phone charger, whereas the wire is this heme, and the protein basically acts like an insulation around this wire. And this is what allows them to move electron continuously over micrometer distances. And we have so far found few outer membrane cytochromes, so we call them OMC, and they have different names like S, Z, and they are so far found to form nanowires. And surprisingly, the same bacteria can make two completely different kind of wires. And here we have measured how fast electrons are moving into this wire by measuring electron conductivity. And the wires which we call OMC-Z are 1,000 times more efficient than the one which they make in a natural environment called OMC-S. And we recently also identified that it's just not Geobacter which make these wires, but many different bacteria and archaea are also producing similar wires. So here I'm showing you the Geobacter species in red, and these blue are archaea, which are involved in producing methane or consuming methane, and there are other bacteria which are not related to Geobacter. And the same is true for this OMC-S wire, whereas phylogenetically very distinct species which are not related evolutionary, they all shows these wires, so they could be widespread, and we are trying to understand how can we use them. So one of the thing really unique about these bacteria that they can form biofilms which are 100 times their size, and they can still transfer electrons from top of the biofilm all the way to the bottom. So like in human terms, I'm 1 meter, and I cannot breathe oxygen, which is like 100 meters away from me. But this bacteria can breathe their electron acceptor over 100 times their size using these nanowires. And this allows them to basically survive in an environment where there is no electron acceptor nearby. And our HFSP project really brought together a lot of my heroes, which I always wanted to work with. My background is in physics, and I'm studying the cryo-electron microscopy structures of this wire, but also to understand functions and how electrons move into them. And my colleague, Lisa Craig, who is a world expert on this pili filament, pili in hair, and for last 20 years, these nanowires were thought to be pili. And we have found that they actually act like a piston to secrete this nanowire. My another colleague, Olivera Francetic, who is a world expert in this secretion system, and she's putting these wires in other bacteria to understand what are the minimal components required to make this wire, and this way we can also identify in which other microbes these wires are functional. And my colleague Carlos Alguera, who is here with us, he's a world expert in understanding electron exchange between this multi-heme cytochrome, and he has pioneered a method based on NMR, nuclear magnetic resonance, and he has discovered how these bacteria transfer electron from inside to these wires. So the 3 questions I want to quickly focus in this short talk is, most C-type cytochromes cannot polymerize into a wire. So what is so unique about these microbe and these proteins that they can assemble into the wire? What's the mechanism? The second question is, it's not sufficient just to have wires on the surface. You still have to get electrons from inside to the outside. So how those electrons get exported from the outside to the inside? And finally, can we actually use different stimuli to control the conductivity of this nanowire, to in turn control the behavior of bacteria, so that we can control how much methane is released to this atmosphere? So let me start with the first part, where a hypothesis we are testing is led by my student Aldo, that the pili, which were thought to be a wire for last 20 years, are actually act like a piston and they remain inside the cell. So pili in Latin means hair, and most bacteria do form pili, and you will hear a talk after me about one of those kinds of pili. But in this particular bacteria, what we found, that pili actually remain inside the cell, and they are involved in basically pushing this wire out. And that explains, over 5,000 papers published last 20 years, where if you do anything to this pili, you don't get these nanowires on the surface. And to test this, we are using genetics and a cryotomography. So this is a work by my student Aldo, where he found that the wires are made up of a unique combination of 2 different systems. So what he found is, if you delete a secretin called PILQ, which is a hole in the membrane, which is used for type 4 pili, there is no significant impact on the bacteria. But if you delete a different secretin called GSPD, which is basically a hole in this outer membrane, the bacteria cannot grow and they cannot make nanowire. So basically, it's the first example that microbes are using 2 completely different machinery together to produce these wires. And we collaborated with my colleague Jun Lu to understand, can we actually visualize the structure of this wire? And we were able to use cryo-electron tomography, which further confirmed that these wires are a hybrid system. So this is an image of a bacteria it's producing the wire. And the yellow arrow which you see is the machinery which we were able to visualize with the tomography. And we were able to identify both the pili and the secretin on the surface. So I'm showing you side and the top view. And what we see is when the pillar remain inside, the gate is closed, and when pillar acting as a piston, the gate opens. And we were able to obtain a structure of that secretin by using subtomogram averaging. And that confirmed that the top part of this system is indeed made up of a type 2 secretion system, whereas the bottom part is made up of type 4 pili. So somehow this bacteria hijacking a component of one system and the other system to assemble these nanowires, and we are further testing that. A key thing is to understand what are the minimal parts we need to assemble this nanowire, and what we cannot create, we cannot understand as a code by Feynman. And inspired by that, what we are trying to do is reconstituting all these systems into an E. coli and trying to see if can we recreate what nature is doing. And my colleague Olivera Francetic has 3 people working on 3 different systems, type 2, type 4, and the nanowire gene cluster to reconstitute this system to figure out what are the parts actually required to make this wire. Another discovery by my student Aldo did is this pila have a cap protein called PILY1, and when he deleted that, bacteria could not make wires or they couldn't transfer electron. And this is a model, AlphaFold model we have built on how this cap protein actually looks like. And my colleague Lisa Greg is studying that. And this is again AlphaFold model of, you can appreciate many different proteins come together just to make a cap. And what we are doing is we are again reconstituting all these proteins into E. coli, and we have been trying several different approaches. And this is the work led by my colleague Lisa Craig. And the next part is how do electron actually can go inside from the outside. And this is the work led by my colleague Carlos, where the question we are really trying to understand is what are these connecting plugs which allow this electron to go from inside to the outside. And Carlos, who is here, and he will present a poster on Wednesday, what his team has discovered using NMR, that there are proteins inside the bacteria, which we call PPC or periplasmic cytochrome. They can directly bind to this wire and transfer electron to the wire. And we just published this work earlier this year. So please stop by that poster. And the last question is, now we know that there are wires and electrons do get to this wire. Can we use that to control the behavior of bacteria? And what we have discovered so far is you can excite this wire using electric or magnetic field or a light, temperature. And in all of these actually do change the conductivity of this wire because electrons move at a different speed, and that has direct consequence to how the electrons are exchanged between bacteria and how the bacteria are growing. So the goal of our team is to really direct this electron flow, and depending on whether electrons are going from left to right or right to left, what we observe is the same microbe either produce methane or break down methane or consume methane. And that's direction we are trying to control to reduce the atmospheric methane level. So with that, I want to summarize the key finding that the work by my colleague Carlos, we were able to identify these electrical plugs which connect the inside of the bacteria to these wires on the surface. We have also identified that these pili, which were thought to be wires for last 20 years, are act as a piston, and it's a hybrid system of 2 different secretion systems coming together to assemble this nanowire. And one of my biggest surprises is these wires are made by bacteria and archaea, which have no evolutionary relation to each other. And we are trying to understand, what are the actual functions these microbes are using this wire, and what is the machinery involved with that. And with that, I want to thank again HFSP for their support to make this project possible and to bring this team together. And thank you all of you for your attention, and please stop by posters. Thank you very much.
Thank you. Questions?
Congratulations, this fascinating talk and teamwork and everything, all HFSP, congratulations. So my question is about your last point. I mean, this is of course extremely attractive concept. Can you elaborate a little bit more? How would you do that? at the end? Because of course, at a scale which would matter, how would you do? How would you change the electron flow at a scale of the wetland or the scale of the cow's stomach?
Right.
So what is he thinking there?
Right. So let me repeat the question for everybody, which is a really important question, that how can we do this at a larger scale? And what we found is we have been using this bacteria to remove uranium from a field site. And the most favorite food this bacteria have is basically vinegar. So when we inject vinegar in the ground site, the Geobacter actually grows more predominantly. And that's what we have been used in the past to remove the uranium from a field site where nuclear plants are. And now we know, under what condition they do make this wire. So our goal is to really use the field site experiment to control, the growth of this bacteria, and then they can partner up with this methane-consuming bacteria. But yeah, that's definitely a challenge to maintain the ecology of the environment and still get the job done. Thank you very much. Yeah.
Okay, it's very fascinating. So I'm curious, when there is oxygen existing there, how stable are the electrons on the nanowire? Will they leak to form superoxide or other rocks?
Right. So the question is how stable the electrons are in the nanowire. Do they react with the environment? And the answer is they're very, very stable because these wires are essential for the survival of the bacteria. So if you remember the structure I showed you, the hemes are at the center, and the protein acts as an insulation around this wire. So electrons are very carefully confined only between this heme molecule so that they don't leak to the atmosphere or they don't react to the atmosphere. So we don't see any evidence of any superoxide forming in these wires. Thank you.
So if you express and purify OMCZ, does it spontaneously polymerize, or does the structure that you've seen by cryo-EM and tomography, is that explicitly dependent on the secretion process in order to get assembled?
Right. So the question is, how do these nanowires polymerize? And as I pointed out, most cytochrome C do not polymerize. They are not capable of polymerizing. What we did found is if we actually take the purify the wire out and break them into a monomer, they do reassemble into a filament spontaneously. So the actual assembly process is just self-assembly, self-polymerization. The challenge is how to actually bacteria get these from inside them to the out, and that's where we think the secretion system plays a role. But you are absolutely right. Once they are outside, they can self-polymerize by themselves. Yep. Thank you for your question. Yeah.
Hello. This is a question more about the ecological strategy. So you get the electrons out, but to really make it work, there needs to be a sink on the other end, right? So do you see any evidence that there's specificity, that sort of the tips of the wires know where they need to go, or that they attach to the right bacteria?
Right, that's a great question. So the question is, electron has to go somewhere. So how do bacteria know where to dump the electron, and what's really how do they figure that out? So in the environment, bacteria use these wires to bind to rocks or minerals or the other bacteria. And I do think the driving force is really the voltage difference between the reduction potential of the rock and the bacteria itself. And we don't know how do they actually sense it, but somehow bacteria do make these wires only when they need it. And we do see that in the laboratory, if we apply this voltage, we mimic the voltage, that actually stimulates the production of wire. So we call it electrogenetics, just like optogenetics. And we are trying to understand the mechanism behind how this electrogenetics work. Yep. Thank you.
This is more motivated by an analogy with neural networks. So do these wires form meshwork, first of all? And can they be used to communicate with each other?
Right. So the question is about how much they are related to neurons and do they form network for communication. So the way neurons work is all about ions, whereas this is all about electrons. So if you see how the neurons work, the insulation layer, which is called myelin, actually controls how much the ions going to move and where they're going to start. Here, actually, the insulation layer protects them. And that's why the electron and ion function is, the mechanism is very different. But we do see that they use these wires for communication. So when this one bacteria is partnering with other bacteria, that's the way they actually form the network. And that's the way they are able to make electron exchange over a longer distance. So it's an analogy, but without using ions, but using electrons. Thank you.
Okay, thank you again.
Thank you very much.